focuses on developing a realistic three-dimensional model of the cervix to study chlamydia and other sexually transmitted infections, or STIs. Chlamydia trachomatis is the most frequently reported bacterial STI in the United States and also globally. The counter here shows almost in real time the number of Chlamydia trachomatis infections during this presentation. Despite available treatments, chlamydia often goes undiagnosed, leading to serious complications, including pelvic inflammatory disease, infertility, miscarriage, and preterm birth. To develop better treatments and prevention strategies, we need a better understanding of how the pathogens interact with the human body. However, current research models they have some limitations. For instance, the most common model used in the lab is based on immortal cells isolated from a cervical cancer tumor in the 1950s. Although this model has been extremely useful for biomedical research, it has certain limitations. For example, these cells do not represent a healthy cervical tissue. There are other models such as primary cells and explants which are more relevant, but they also lack some things. For instance, they have a limited lifespan, and they also are hard to standardize because they are different between different people. Animal models, while offering a multicellular complexity, they do have physiological differences as well. For instance, the animal menstrual cycle is different than the female menstrual cycle, and the bacteria that lives in the reproductive tract is also different, and this is known as a microbiota. To develop better treatments and prevention strategies, we need a better understanding of how these pathogens interact with the human body, and therefore we are basically in collaboration with our partners here at UVA, at the University of Maryland and the University of Delaware. We are developing a realistic three-dimensional model of the human cervix, one that can basically mimic physiology and also architecture, as you can see there, and other things like the microbial environment. With this model, we'll be able to mimic more closely the end of the cervix. This model is based on a silicon device, a silicon-based device with microfluidics, two microfluidic channels separated by a permeable membrane, as you can see here or on the slide. This allows us to grow multiple cell types, basically mimicking architecture, and to simulate also fluid flow, which is common in certain organs such as the female reproductive tract. And this is done by connecting the insert to a low-flow peristaltic pump. So far, we have been able to infect this model with chlamydia, with a fluorescent chlamydia strain, as you can see in this image, where each dot represents a chlamydia basically, a chlamydia inclusion. What's next? We want to increase the complexity of the model by introducing extra components, cellular components and non-cellular components, and also the microbial environment, to basically answer key questions that haven't been answered yet. For instance, the role of female sex hormones in Chlamydia trachomatis infection, as well as the role of the immune response to chlamydia and other sexually transmitted infections. The counter here again shows that over 800 chlamydia infections have been reported during my talk. And finally, I would like to invite you to my poster if you have any questions or if you're interested in using this model by any chance. Thank you very much.